Okay, we have hit the top of the hour. My name is Adam Bogdanov. I currently serve as the president of IS-MPMI. And on behalf of the board and the organizers and the speakers, moderators today, I wanted to welcome everyone to the 2025 IS-MPMI Early Career Showcase. To some of you, I say good morning. To some of you, I say very early good morning. Some of you, good afternoon, and some of you, good evening. Some of you even actually late evening. So thanks for being with us. We have a terrific lineup of speakers today, and I'm really excited to see that so many of you have joined us. Moderating for us today are Linan Jie. And Linan, can you wave?
Yeah, hello everyone.
Yeah, Linan is a postdoctoral scholar at the CAS Center for Excellence in Plant Molecular Sciences. And joining her as co-moderator is Yumino Sasaki. Yumino, say hi.
Hello.
Yeah, Yumino is a grad student in plant pathology and plant microbiology here at Cornell University. And very quickly, let me introduce sort of the director of the show here, Katie. You'll hear from Katie later in the program. Katie has helped organize, and I want to thank her for that. And she'll also be in charge of setting up the breakout rooms for the discussion session at the end of the presentations. Katie? Yep, great. Okay, before starting though, and let me just quickly review the plan for today. We'll have 6 talks, and we'll do these in blocks of 3. So after the first 3 talks, we'll have a 10-minute break, and then we'll have the remainder of the 3 talks, and then we'll go straight into the breakout discussion. And the breakout discussion topics will be posted during the break, so you get a chance to think about which room you'd like to join. We ask that, as many of you have already, we keep our video off during the presentations to help with bandwidth for those who might have narrow bandwidth with their internet connections. But for the Q&A for each talk, feel free to turn your video on, raise your hand, or use the chat window. So the moderators will monitor both the chat window and anyone who's raising their hands. And if they call on you, then you can unmute and turn on your video for no problem there. I think that's it. Unless there's anything I might have forgotten, I'll ask Yumino, Linan, and Katie, was there any point that I missed?
I think you hit it all there, Adam. So
okay, great. So
let our moderators.
Yeah, without further ado, let's move on, and I'll hand it over to you, Yumino and Linan, and yeah, enjoy the talks.
Okay, let's just start. Our first speaker is Xiaofei Zhu, who is a doctoral student at China Agricultural University. She will be presenting on interfamily co-transfer sensor and the helper NEORAS extends immune receptor functionality between adjuvants. And by the way, she loves kitty very much, and the most common photos in her smartphone is kitty. So please enjoy. So can you show your screen?
Okay, thank you for your introduction. I will start my presentation. Everyone, my name.
I'm sorry, we can see.
Can you see my screen?
We can see.
Okay.
No.
Hello, everyone. My name is Xiaofei Du. Thank you to the board committee for giving me this opportunity. I'm honored to present my research project. And I'm a PhD candidate at China Agricultural University under the guidance of Professor Hailong Guo. Our lab studies plant immunity using 3 spatial systems: Arabidopsis, Pseudomonas syringae, rice magnetoposa arrays, and rice Pseudomonas arrays, aiming to uncover the mechanisms of plant immunity and develop strategies to enhance host resistance against both fungal and bacterial pathogens. To begin, let's briefly introduce plant immune receptors and their deployment in crop breeding. Plant pathogen cause severe disease, threatening global crop production. Plant defense against microbial infections are initiated on perception of pathogen-derived molecules by cell surface localized PRRs and intracellular NLRs. Cell surface localized PRRs detect apoplasmic pumps and initiate partner-triggered immunity. Adapted pathogens secrete effector proteins into host cells that promote virulence. Plants has evolved intracellular NLR immune receptors that detect effectors and activate effector-triggered immunity. This cartoon illustrates two key strategies for developing disease-resistant crops. One is inter-family transfer of immune receptors to expand pathogen recognition capacity. Another is engineering immune receptors to enhance disease resistance using multiple strategies. Today, I will focus on the interfamily transfer of immune receptors. And as I mentioned before, plant PRRs recognize conserved pumps, offer broad-spectrum resistance. Engineering disease-resistant crops using PRR is a promising strategy. The brassicacid-specific cell surface localized receptor-like kinase EFR recognize the conserved bacterial ligand ELF18, transferring EFR from Arabidopsis into Nicotiana, benzimiana, and tomato, confers resistance to Pseudomonas syringae-induced angular leaf spot and Rostonia solanacearum-induced bacterial wilt in these transgenic plants. If EFR transferred into monocoat rice, the transgenic rice exhibit enhanced resistance to XOO, which causes bacterial blight disease. And the transfer of RP23 from Arabidopsis thaliana into tomato enhanced resistance to omicid-induced late blight and fungal-caused sclerotin stem root, representing another successful example of crop species PRR transfer. There are also successful cases for intracellular immune receptors. One example is WRR4 from Arabidopsis thaliana confers a Beagle candida caused white rust resistance in oilseed brassica crops. WRR4 encodes a cytoplasmic TNL that confers a dominant white rust resistance. The cotyledons of WRR4 transgenic plants with showing resistance phenotypes, no formation of pustules. Another example is transferring pepper BS2 gene into other crops to enhance disease resistance. The BS2 recognizes AVR BS2 effector from the pathogen XCV, which causes bacterial spot disease, while it transfers into tomato, successfully suppress XCV growth. However, similar transfers into Arabidopsis and cassava failed to provide resistance. The phenomenon was referred as restricted taxonomical functionality, and many NLRs exhibit RTF, meaning they only can function in closely related species. XOC is another Xanthomonas species. XOC occurs bacterial leaf streak in rice, a major staple food in China, posing a significant threat to agricultural productivity. To date, no endogenous BLS-resistant genes have been identified in rice. Our lab is focused on this issue, and given that AVR-BS2 is highly conserved among Xanthomonas species, we are exploring whether the BS2 gene can be used to improve rice resistance to XOC. Firstly, we tested whether the pepper BS2 receptor could recognize AVR-BS2 in XOC. We performed transient expression assay. In Nicotiana benzimiana, AVR-BS2 from XOC can reduce hypersensitive response when co-expressed with BS2, suggesting that AVR-BS2 of XOC is also recognized by BS2. Then we tested whether BS2 transgenic rise confers resistance to XOC. However, no resistance was observed in BS2 transgenic rise against XOC, indicating that the pepper BS2 gene has restricted taxonomic functionality and failed to function in rice. An emerging paradigm in plant immunity is that many sensor NLR require a partner NLR or helper NLR to mediate immune activation and disease resistance. Sensor NLR mediate pathogen perception and activate downstream helper NLRs, which convert recognition into immune activation. In Arabidopsis, helper NLR, ADR1, and NRG1 are required for full function of TNLs and several CNLs. Similar to ADR1 and NRG1, several NLR required for cell death, called NRC, are necessary for cell death and resistance activities of multiple solanacea sensor NLRs. But NRC helper NLRs are only found in asteroid and are absent from monocots. This dependency suggests that transporting both sensor and helper NLRs together might enable resistance in non-host plants. To address this, we explored whether the co-transport of sensor and helper NLRs could overcome RTF. Now let me show our experimental results. We generated transgenic rice expressing BS2 with NRC2, NRC3, or NRC4, as well as a combination of the NRC2, 3, 4, and tested whether transgenic rice could confer resistance to XOC. Rice lines co-expressing BS2 with NRC3, NRC4 were fully resistant to XOC. No lesion observed after inoculation, and bacterial growth was significantly reduced in these lines compared to wild type, suggesting that XOC became virulent on these transgenic rice plants. Importantly, the AVR-BS2 mutant strain of XOC, which the AVR-BS2 gene was disrupted, was still able to cause disease in all rice lines, confirming that resistance was AVR-BS2 dependent. A critical concern when stacking immune receptors is whether it negatively impacts plant fitness or basal immunity. Next, we evaluate the impact of stacking sensor and helper NLRs on agronomic traits and basal immunity. As shown in this slide, the wild-type and transgenic rice lines exhibit no significant difference. Statistical analysis revealed that plant height, tiller number, grain number, and grain weight are all comparable between these transgenic lines and the wild-type line. And transgenic rice lines also didn't show constitutive activation of defense response, as measured by MAPK, cascade activation, and rose production, and in response to pump. Additionally, transgenic lines was not more resistant to the fungal pathogen magnetopause arising, indicating that the basal immunity of these plants was not altered. These findings demonstrate that stacking sensor and helper NLRs doesn't compromise plant fitness. TSL Jonathan-Jones group also demonstrated similar findings. BS2 transgenic lines failed to trigger a hypersensitive response when challenged with Pseudomonas syringe delivering AVR-BS2. In contrast, BS2 NRC co-transgenic lines exhibited a stronger HR under the same conditions. Additionally, co-expression of BS2, NRC, and AVR-BS2 in cell being protoplast cause the cell death and lead to reduction in the luciferase activity. As demonstrated, the BS2 gene can function when transferred within the Solanaceae family. However, its transfer to distantly related species fails to confer resistance. This limitation can be overcome by co-expressing BS2 with NRC, enabling its functionality during interfamily transfer. This strategy could enhance resistance in crops such as cassava and brassica. Since monospecious XAM cause bacterial blight in cassava, while XCC cause black root in brassica, by redeploying the BS2 and NRC genes may also revolutionize resistance to this crop disease. And Phytophthora is a highly destructive pathogen that causes devastating disease in many plants. This pathogen secretes RxLR effectors, and some RxLR effectors are conserved, such as AVR-AM3 from P. infestans, which is recognized by the RPI AMR3 in Solanum americanum. Importantly, RPI AMR3 can recognize AVR AMR3 across multiple Phytophthora species. Therefore, co-transferring sensor and helper NLRs shows great potential for engineering resistance against Phytophthora in vulnerable crops such as strawberry, cacao, and soybean. This strategy addresses the lack of natural resistant genes in these crops and offers a promising solution to combat Phytophthora-related species. And lastly, I would like to thank my supervisor, Hailong Guo, and my lab members, and our neighboring lab member for their support. Thanks to our collaborators, Jonathan Jones, and their lab members. And we also thank Professor Sofen Kamen for sharing classmates. Thank you for your attention, and I'm happy to take questions you may have.
Okay, okay, thank you for the interesting. Okay, we see that we can see some people. Oh, please. Adam, I see your hands up. So,
I was just applauding. That wasn't asking a question. I'll defer to others for
oh,
questions.
I'm sorry. Sorry
No,
for
that's
the
okay. That
misunderstanding.
was confusing on my part.
Okay. And okay, so does anyone have the question? You can just hands your okay, the Jean, please.
Very nice talk and very exciting. I'm wondering if you talked about you used the word stacking because you're using two genes together, but there's still the worry that the pathogen may lose AVR, the AVR gene that is matching the BS2 specificity. So is there any idea to further, transform the rice with another R gene so that you don't have a single gene vulnerability
Yeah.
to lose that? Do you get my point?
Yeah, thank you for your question. In our experiment, we combine the BS2 and the helper NRC into rice and found they because BS2 need NRC to function. And you asked some other single question, other single NLR genes can transfer between different species.
not exactly. I'm thinking that if you only deploy BS2 in rice with its helper, the pathogen still may lose the effector that's recognized. Like it's only one gene in the pathogen to overcome resistance. So we had the concept of stacking, where you would have multiple resistances there, so that in one step you couldn't overcome.
I get the point.
Yeah, so I wondered if you were thinking along those lines.
Yes, because the resistance is as we can see, it's full resistance to XOC, and this gene push pressure on pathogen evolutionary, so it's easier to overcome by pathogens. To address this question, stacking many NR genes into other groups is a useful function, is a useful method. I think if this transgenic lines used in field, it's better to stack multiple NR genes in order to avoid overcome by evolutionary pathogens.
Okay, maybe one more question. Mauricio, can you? I'm sorry, maybe I don't know how to. Yeah, please.
That's okay. Yeah. Hi, Xiaofei. Great talk. I guess my question is related to basically you transform BS2 into rice and you deliver either NRC2, NRC3, NRC4, or all three NRCs. So I guess you didn't really see any difference between adding one helper and several helpers, right? Or did you see any differences? Was BS2 working better with some helpers, with one of the helpers and not with the others? And moving forward, for example, for your cassava work, are you planning on co-transforming the sensor with one helper or with multiple helpers? I was wondering if you could comment on that. Maybe it was too many questions at the same time, sorry.
Thank you for your question. Yeah, we transferred the BS2, NRC2, NRC3 into rice. They, especially NRC3 and NRC4, exhibit full resistance. And if we trans this gene into cassava or into brassica, there we may have results to prove that BS2 NRC transgenic cabbage may have resistance to XCC.
Okay,
So sorry.
time's up.
Yeah, never mind. Sorry.
Maybe you can use the chatting for the future discussion. Yeah. And okay, I will just move to next speaker. Our next speaker is Dingliang Zhang, who is a postdoctor, and he likes tennis and badminton. And he also comes from China Agricultural University. He will be presenting on the UBR-mediated turnover of AL7 functions loss accumulation in AM-mediated immunity. Okay, please share your screen.
Okay, thanks for your attention. No, but thanks for your introduction. Yeah. Hello everyone. I'm Dingliang Zhang from China Agricultural University. Thanks for MPMI for giving me this opportunity. It's my great honor to present my work to here. The focus of my presentation is UBR7 mediated turnover of L7 fine-tunes loss accumulation in immediate immunity. Let me start by introducing myself briefly. I'm Dinliang Zhang from China Agricultural University, currently a postdoctoral researcher in Yongliang Zhang group, and I'm focusing on plant antiviral immunity. I have divided my presentation into the following 4 parts. First, let me introduce the related background. Plant viruses pose a serious threat to agricultural production and food security. It's the most economic and effective manner to cope with plant virus threat by application of viral resistance or susceptibility genes. Plant innate immunity comprise pattern-triggered immunity and effect-triggered immunity. PDI is mediated by plasma membrane localized pattern recognition receptors, and the ETI is mediated by intracellular resistant proteins like NIRs. N-gene from Nicotiana glutinosa is the first cloned antiviral NIR gene. N protein confer resistance against tobacco mosaic virus by recognizing the helicase domain P50 of TMA, and then elicit a hypersensitive response to restrict plant virus in the infection region. N-TMA interaction has long been used as a model for studying plant antiviral immunity. And during immediate resistance against TMA, some host factors have been identified to participate in this process. However, the low abundance and highly flexibility of NIRs makes it difficult to identify the other interactors. So we developed TBID-based proximity labeling in plants, which has advantages in identifying weak interactions or transient interactions, and we used it to identify the proxy term of N protein. From N protein proxy term, we select 2 possible interactors for further investigation. The first one is UBR7. UBR7 is a putative E3 ubiquitin ligase, belongs to UBR box protein family. Our result suggested that UBR7 interacts with N protein and promotes its proteasomal degradation. Interestingly, TMA factor P50 is able to interfere with the interaction between UBR7 and N protein to stabilize N protein. Another possible interactor of N protein is a transcription factor, L7. Alphin-like family proteins are known to be a kind of unique transcription in plants and have been revered to regulate seed germination or responses to abiotic stresses. In our study, we found that L7 interacts with N protein directly and positively regulates immediate immunity. To revert the underlying mechanism of L7 in regulating plant immunity, we proved that L7 binds to the promoters of rose scavenging genes, APX2, GR, and GPX2, so that and repress their expression, so that promoting rose accumulation and inducing cell death. Furthermore, we also found that MAP kinase SIPK and WIPK can phosphorylate L7 at serine 174. And this phosphorylation enhances the DNA binding ability and the transcriptional repression activity of L7. In resting state, low phosphorylation of L7 allows the expression of rose scavenging genes to maintain rose homeostasis. Upon TMA infection, activated MAP kinase phosphorylate L7. Phosphorylated L7 repress rose scavenging genes to promote rose accumulation. But we all know excessive rose production can be toxic to plants. So they have evolved mechanism to restore Ross homeostasis once the pathogen infection is attenuated to avoid fitness costs. So we wonder that whether and how plants fine-tune the L7's promotion of Ross production. And what's the relationship between the two components of N protein, UBR7 and L7? We next conducted a series of experiments around these two issues. During TM infection, L7 protein level decreased gradually. To further examine whether this reduction is associated with ubiquitination, we next detect the ubiquitination of L7 in vivo. And the result of cell-free degradation assay showed that L7 is an unstable protein. This result suggested that L7 undergoes ubiquitin-mediated degradation during immediate immune response. To identify the E3 ubiquitin ligase mediating the degradation of L7, we utilized TBID-based proximity labeling to analyze L7's proximity. And we found that E3 ubiquitin ligase UBR7 is identified in L7 proximity. Consistently, we also identify L7 in UBR7 proxy term. We further confirm the interaction between UBR7 and L7. And we next start whether UBR7 degrades L7. We found that UBR7 co-expression decreases L7 protein level, which can be restored by MG132. In UBR7 knockdown transgenic plants, L7 ubiquitination level significantly decreased compared to that in NN plants. And by in vitro ubiquitination assay, we next confirm that L7 is a direct substrate of UBR7. To identify the ubiquitination site of L7, we next perform the IP-mass. The result of IP-mass and the ubiquitination level analysis demonstrated that lysine 20 is the key ubiquitination site of L7. And UBR7 had no pronounced effect on the protein level of K20R mutant. To clarify the functional relationship between UBR7 and L7, L7 was transiently expressed in NN and UBR7 knockdown transgenic plants. We noticed that UBR7 knockdown promoted L7-induced cell death and enhanced Ross accumulation. Moreover, L7 K20R mutant induced stronger cell death and higher Ross levels compared to wild-type L7. Above result suggested that UBR7 negatively regulates L7-induced cell death and Ross accumulation by promoting its degradation. We have revealed that TM infection induced L7 protein level decrease. To further investigate whether immune activation facilitates L7 degradation, we introduced P50 and MEK2DD to activate plant immunity in NN plants. We found that both P50 and MEK2DD can reduce L7 protein level in proteasome-dependent way. P50 and MEK2DD can activate MAP kinase SIPK and WIPK to phosphorylate L7 at 174. So we guess that S174 phosphorylation is possible to affect L7 stability. So we further compared the protein level, stability, and ubiquitination level of L7, S174A, and S174D. We found that phosphomimic mutant S174D had less protein level compared to L7 and S174A. And the result of cell-free degradation assay showed that S174D degrade more quickly than L7 and S174A. Besides, the ubiquitination level of S174D A and S174D were decreased and increased than L7, respectively. This result leads to the conclusion that S174 phosphorylation of L7 can promote its degradation. So we wonder that whether UBR7, whether E3 ubiquitin ligase UBR7 is required for S174 phosphorylation induced L7 degradation. To answer this question, we next analyzed the accumulation of L7 protein level in both NN and UBR7 knockdown transgenic plants during immune activation. The results showed that UBR7 knockdown reduced the degradation of L7 induced by immune activation. Consistently, the protein level, stability, and ubiquitination level of L7, S174A, and S174D were comparable in UBR7 knockdown transgenic plants, implying the requirement of UBR7 for reduced stability of phosphorylated L7. To explore how L7 phosphorylation facilitates its UBR7-mediated ubiquitination and degradation, we used the online AlphaFold server to predict the complex structures of L7, S174A, S174D with UBR7. The analysis result suggested that S174 phosphorylation increased the number of hydrogen bonds between L7 and UBR7. And the result of split lag, CoIP, and MPP pull down further confirms that S174 phosphorylation enhanced the interaction between UBR7 and L7. Our findings lead to the proposed model. In resting state, UBR7 degrades N protein, and the low phosphorylation of L7 lowers ROS scavenging genes expression. Upon TM infection, activated MAP kinase phosphorylate L7 to promote ROS accumulation. Furthermore, phosphorylation of L7 enhanced the interaction between L7 and UBR7, leading to UBR7-mediated ubiquitination and subsequent degradation of L7. This degradation restores the expression of ROS scavenging genes, preventing excessive ROS accumulation. At last, thanks for the guidance from my supervisors Yongliang Zhang, Dawei Li, Savis Rama, Dinesh Kumar, and Minliang Xu. Thanks to my collaborators Xinyu Zhang and Yiping Wang for their effort on the work progress. Thanks for the support from National Science Foundation of China. Yeah, okay, and that's all. Yeah, thanks for your attention. That's all.
Okay, interesting presentation. Does anyone have comments or question? You can hands you up or just use the chatting window. Okay, and I have a question. So, in your final model, can you show your final model? So you found that the E3 ubiquitin ligase can degrade AL7, and this ubiquitin E3 ligase is also activated by the immune signaling.
Yeah.
So why plants have this kind of response? I mean, when the immunity is activated, why it's want to AL7 degradation? I mean, is that a kind of
Yeah, that's a good idea. That's a good question. I think the degradation of L7 is aimed to balance plant immunity and plant growth. because we know over immunity can cause a fitness cost of plant growth. So while the passenger invasion is attenuated, it is necessary to keep the plant immunity to a low level and make the to allow the energy for plant growth.
Okay. Oh, and Jean have the question, please.
Oh, yeah, I am interested in not only the idea that you might have ways to turn on and off or modulate the ROS as you have discussed, but also the potential spatial distribution of where these events are taking place. For example, I know that those viruses sometimes make kind of a lesion, and I wondered whether are all these events happening in all the cells, or do you think they're happening around the lesion, or where might they be happening in the infection zone?
在它们在坏死斑中、坏死斑还是在坏死斑周围会发生，然后讨论这个
Yeah, this
question.
it's a good question. In fact we considered before. We think maybe as you mentioned, maybe the degradation of IL-7 can occur around the cell death region, but we don't have the direct evidence, not yet. Yeah.
Good question.
Yeah, that's a very good question. Yeah.
And also it's have systemic acquired resistance. 系统
Oh, thank you.
Okay, we still have time, so maybe one more question. Okay, so I have another question. So, have you identified the upstream of UBL7? So I think the phosphorylation of AL7 and the ubiquitination of AL7 maybe is activated in two pathway. So is that in two pathway or just when plants responds to the N the immunity signaling, so phosphorylation, the positive of promote AL7 and the ubiquitination of AL7, how plant manage it. So in your model, you're showing that loss accumulation is activated by AL7, and the loss also may be affected by AL7 ubiquitination. They have two upstream. Maybe related to my first question, so I'm still interested in the upstream of the KNS and also the E3 ligase. So
目前的3它这个在它自然酸化以后再激活这个BR7。
In fact, the evidence we haven't yet proved that UBR7 can degrade phosphorylated L7. Yeah.
Yeah, okay, we can discuss later. Anyway, time is nearly up, so let's just finish this part and maybe move to next speaker.
Okay, thank you.
Yeah, thank you so much for your presentation. And our next presenter is Masuro Ezekiel Harris. I hope I speak right. So he is a He is the doctorate student at INGB Coincident. He will be presenting on genomic and transcriptomic profiling of Argentinian Phytophthora infectans isolates to diprolate bright resistance in the region. And he loves video game, and me too. So anyway, please enjoy his presentation. Yeah, you can start. Sorry, maybe you mute yourself. We cannot hear your voice.
Are you hearing me now?
Oh, yeah, I can hear you now.
Okay, sorry. Good morning, everyone. Good afternoon to some of you. Today, I'll be presenting some of my PhD work, understanding Phytophthora infestans in Argentina from population structure to resistance strategies. My name is Marcelo Juarez, and I work at INGEBI CONICET in Buenos Aires, Argentina, under the direction of Dr. María Eugenia Segretin. I'm doing my PhD at the University of Buenos Aires, and I work on plant pathology and bioinformatics. Today, we're going to be talking about Phytophthora infestans, an homicide and causal agent of the potato late blight. It evolves rapidly, in part due to its genomic architecture, where you can see these red spots are virulence genes, which are located in gene-sparse regions with lots of transposons activity, repeat-rich regions, and things that help to turn on and off these effector genes. Now, in plant pathogen interactions, effector genes are often recognized by immune receptors. There is going to be a lot of talk about immune receptors here. We are only focused on the phenotype of resistance. And in the case of Phytophthora infestans, there has been many immune receptors cloned from wild Solanum relatives, and we'll be using some of them to test their potential to bring resistance to Argentinian potato cultivars. So potato production in Argentina is around 2.5 million tons, and the Punta cultivar is the most spread one, as it is 60% of the crop area in Argentina. And other relevant cultivars include Kennebec and Innovator will be working with these three. So in this presentation, we are going to address three main topics. First, we are going to characterize the population structure of infestans in Argentina using two different approaches. We are going to assess Argentinian isolates potential to infect these agronomically relevant cultivars. And then we are going to evaluate some selected R genes and conferring resistance against these local isolates. So we obtained 20 isolates from Argentina's major potato producing areas, and when we genotyped them by their SSSR profile, this is a 12 SSSR that if someone here works on Phytophthora population studies, they might know it because it is widely used. When we genotyped our isolates, we found that all of them belong to the Europe 2A1 multilocus genotype here on MLG. And this MLG, as it was described in Europe, and it had a bigger importance there in the 90s and early 2000s, where it showed more prevalence. But right now, in the last 10 years at least, in the countries marked in yellow here, it was reported, although it showed a very low prevalence. Where it was with high prevalence are the countries marked in purple, so it seems that it's more of a southern hemisphere problem right now. And although this SSSR profile allowed us to identify our isolates as part of this MLG, it showed very low diversity within the population. So we explored a bit more of that with an Amplicon-seq panel. I'd like to thank Kurt Lamour here for making us the libraries for this study with his multiplex Monsterplex company. The Amplicon-seq panel consisted of 98 targets of gene fragments up to 300 base pair long. And we used some control samples from the same MLG, but from different countries and different years, and these other two MLGs. This amplicon-seq panel, we are going to be exploring it for SNP data. And when we made a tree with this SNP data, we first could see that the three main branches we obtained in our tree were supportive of the SSSR genotype of the samples. I'd like to thank for all of these white samples to David Cook for providing us the amplicon-seq data and the SSSR genotyping, and Florencia Luca for collaborating with us in making the collection of Argentinian isolates. And within the Europe 2A1 branch, you can see that Argentinian samples could cluster tightly together, indicating clonal population. So given this clonality, we selected one isolate, PINSL, to perform whole genome sequencing and transcriptomic data of the infection of the Spunta cultivar. And we selected other three isolates from major potato producing areas of Argentina to perform infection assays. So this representative isolate showed a triploid genomic architecture that's also indicative of asexual reproduction. As you can see in this histogram, where allele proportion of heterozygous SNPs is shown, and you can see in this by bimodal distribution that it is a triploid genomic architecture. So we also seen the gene loss assessment. We found 21 effectors absent with respect to the reference genome T30-4. But in the next slide, we are going to focus on the loss of ABR1 effector, as the lack of this effector should trigger R1 resistance broken. And we care about this R gene because it is the only one described to be deployed by cultivars Spunta and Kennebec. And as we expected, PINSL was able to infect both of these cultivars and all the other isolates we tested also. So we believe that ABR1 gene may explain resistance breakdown on both these cultivars. It was a bit trickier with innovator cultivar, as we found different ecotypes within this multi-locus genotype lineage towards this cultivar. As you can see, we had some isolates which were virulent and some others which wasn't. And innovator is described to have 3 resistance genes against Phytophthora infestans, R1, R2, and R3b. We already ruled out ABR1 as a candidate, but we don't know what happens with ABR2 and ABR3b in this context. What we do have some data to explore is in the PINSL and Spunta interaction. We performed an RNA-seq of this situation in 3 independent experiments. And during my PhD, I focused on pathogens expression profile and during host colonization. And today we are going to use some of those results to discuss key observations here. We are going to use that because what we've seen in Spunta is that PINSL upregulates ABR2. In fact, it is one of the most upregulated genes in this pathosystem, but we found no real aligning in transcriptomic data to the ABR3b gene, although we had genomic coverage of this. And it wasn't expressed in planta samples or in control plates. So we believe that this gene is silenced here, and an hypothesis we have is that ABR2 polymorphisms at some level, transcriptional mutations or something, we have to figure it out yet. It may be involved in this resistance breakdown. We also worked with transgenic Nicotiana ventamiana, expressing different immune receptors to confer resistance to late blight. We already talked about R1. It was expected that PINSL would break its resistance, and in fact it did. as it doesn't express the effector. It was a different case with ABR with R3A immune receptor, as we found expression of the effector ABR3A, but it was an allelic variant EM, which is known to disrupt resistance by R3A, and that's what we also saw. But immune receptors RPI, BLB2, AMR1, and AMR3 showed a strong hypersensitive response in these transgenic lines, and we also seen expression of the cognate effectors of these three immune receptors with different levels of expression. And although this is in spunta, we should try this on the Nicotiana ventamianas, but ABR AMR1 wasn't upregulated during the infection, and we have to elucidate here if this expression level it will be the same in Nicotiana ventamiana, or it is induced, or if this baseline expression is sufficient to trigger immune response. Juan Azcue here during his undergrad final work collaborated in these experiments, and also he sequenced the effectors that are recognized by these immune receptors in the three remaining isolates, and then compared them to PINSL genomic sequences we had, and he found no sequence polymorphisms between the isolates and no resistance disrupting variants in the three effectors that are recognized. And also he didn't found polymorphisms in R3A. So genes AMR1, AMR3, and BLB2 should control all tested isolates. As a take-home message, I would like to be that Argentinian population seem to be dominated by a clonal lineage, that all potato varieties are susceptible to late blight, at least the ones we care about, and that Argentinian isolates have overcome R1 and R3A resistance, possibly R3B, that the differential response in innovator may be explained by R2 breakdown by some virulent isolates, and that I already said that AMR1, AMR3, and BLB2 should all be good candidates to deploy resistance in these varieties. This is our phytopathology group. Maria Eugenia Segretin is our PI, and I'll be working with her during my postdoc on this ecotype differentiation within the Argentinian clonal lineage. Mora Guaglianone is working on spunta transformation to confer resistance to late blight, and Valentina here is doing her undergrad project on immune receptor profiling of spunta. Spunta transformation is it's a comfort zone for our lab, as we have a lot of experience on that, and indeed we have Ticar, the first transgenic potato, approved for sale in Argentina, and it deploys resistance against PBI, which is a problem here. I'd like to thank to the Plant Biotechnology Lab at INGEVI CONICET, and special thanks to Professor Sofia Moon and Jonathan Jones' lab for the Nicotiana ventamiana transgenic lines. I already said thank you to these three people, and Liliana Cano Mogrovejo for the informatic advisor during my early steps on it. I don't know if I was too rushed, but I am ready for your questions or answers.
Okay, thank you so much for your presentation. Oh, yeah, Jane have the question. And Jane first, please.
Oh, I actually was just clapping, but maybe I can think of
Sorry,
something.
I misunderstood again.
Yeah, that was really nice. I was wondering, this is maybe an adjacent question, but how's the acceptance of using the transgenic potato in your area?
I'm sorry, I didn't understand how it was.
Like, do you think the public will accept transgenic potato?
With TICAR, the TICAR experience had it was a long run. It was the PhD of the PI of the whole lab in the 90s, and it was released in 2021 or something like that. It was like 30 years of a struggle with regulatory. Now. Argentina has a potato production that is heavily mechanized, and their potato producers are kind of accepting technologies to improve their crop. And in Argentina, it isn't that hard as it might be in Europe, where I read some things about regulation there. And we have already TICAR, and we are working now with the same company that helped us release TICAR in PBL and PLRV, potato leaf rot virus, and PBI resistant. And PhD from Mora. I don't know if that's a project that I list. I am expecting to be able to transfer that technology to the market too.
Thank you.
Okay, we still have time. Does anyone have the question? You can use the chatting window and also just hands up. I will not make a mistake again. Okay, I have a question. So, why you choose the tobacco as your experiment materials?
I'm sorry.
Why you choose the tobacco? I mean the new blends as your experiment materials? Because I think it's far from the potato.
I'm sorry. I'm having trouble to understand the question. I believe you are asking me why to do the experiments on Nicotiana, or
Yeah, because you use this plant, the transgenic new blends. Yeah, I understand it's easy for making the transgenic plants.
the transgenics, as I thank to Sofia Camuns and Jonathan Jones' lab, we had the RNA-seq we had the RNA-seq, and we had some candidate genes there, mostly their different expression profiles, as I may show here. I'm still sharing, yeah. So we had these differently expressed genes, and we wanted to test all of them out. And Professor Sofia Camun's lab shared with us these transgenic lines. So why did we choose this? Is because that's what we had.
Okay, I understand. Yeah, I understand that is some limitation. I just I thought it will have some. Okay, please, I saw it's a hands up. I see it. Please, Mauricio.
Okay, I know this guy.
Hi, Marcel. Really nice talk. I was wondering, I don't know if maybe you mentioned it and I missed it, but for this, I think it's ABR2 that you mentioned that there might be some polymorphisms that lead to the loss of recognition. Do you know what those SNPs are, and like have you tried modeling them onto like an AlphaFold structure of the effector or something along these lines, or do you don't know yet what the differences are for that effector?
No, we don't know that yet. We are hoping to do some RNA-seq experiments on Innovator and see what with virulent and virulent isolates, and see where is that polymorphism. We also want to get some genomic data for this and compare them. It can be transcriptomic level, it can be mutations, it can be gene loss. We don't know that yet, but.
Oh, okay, perfect. Thank you very much.
Okay, I think time's also up, so we will just finish the first three presentation, and I think we'll have a break break. Yeah, yeah. So
Great,
maybe we
thanks.
will have a rest.
Yeah, thanks very much. That's exactly right, Nanan. Let's reconvene at 15 past the hour. So we have a little time to get up, stretch your legs, maybe a cup of coffee or a snack. And during this break, come back to the screen at some point to check out the breakout room discussion topics. And yeah, we'll see you at quarter past the hour. Okay, welcome back, everyone. Before we move into the next three talks, a procedural note here. I think we're okay with bandwidth. I really do. So for anyone who would like to keep your video on during the talks, speakers can see your faces that way and judge whether they've said something confusing or something exciting. I think that'd be great. So I'm going to leave my video on. Feel free to do the same. And Yumina, over to you.
Great. Thanks, Adam. Can everyone hear me okay? Okay, so before we get started, I just want to reiterate that you are free to use either the chat option or just raise your hand for questions, whatever is easiest for you. So without further ado, I am excited to announce our next speaker, Mauricio Contreras. He is a postdoctoral scholar at the Sainsbury Laboratory. And his fun fact is that he won Who Wants to Be an MPMI Millionaire contest at the last IS-MPMI conference. And then, because of that, he got to sing Happy Birthday to Roger Innes in Spanish in front of all the attendees there. I was one of them, and he was a great singer. So yes, I enjoyed that as well. So yeah, take it away, Maurizio. His talk is titled Structural and Biochemical Basis of Activation of an NLR Immune Receptor Network.
Can you hear me now? There we go. Perfect. Okay, thank you, Yumino, for the introduction. Yeah, my name is Mauricio Contreras. I'm based in the Sainsbury Lab in Norwich, UK. I'm currently doing a postdoc with Sophie and Kamoun, working on plant NLR immune receptor biology and effector biology. So I guess effectors don't need an introduction with this crowd. They are secreted by pathogens to modulate host physiology and promote disease. And then some of these effectors can be recognized by the plant immune system. We've heard a lot about NLRs already. So these like intracellular events of effector recognition are mediated by this NLR class of immune receptors. And then when an NLR recognizes a cognate effector, this leads to NLR-mediated immunity and disease resistance, which is often accompanied by a form of programmed cell death. So NLRs typically have this tripartite domain architecture with an N-terminal signaling domain, a central MBR domain, and a C-terminal loose-enriched repeat domain. Today I'll be talking about NLRs with this N-terminal coil-coil type signaling domain, although you should know that other types of signaling domains do exist. So how do these NLRs that we've been hearing about convert effector perception into immune signaling? So I guess the first paradigm that was established was based on the CC-type NLR from Arabidopsis thaliana, SAR1, so a bit of a model NLR. So in the case of SAR1, SAR1 exists as a monomer in its resting state, and then following effector perception undergoes a series of conformational changes which allow it to oligomerize into this pentameric structure, which was termed the resistosome. So then this resistosome inserts itself into a plasma membrane to initiate immune responses and eventually to trigger cell death. And what's very cool about this is that actually NLRs are not only a component of plant innate immunity, so NLRs are found in the innate immune systems of bacteria and mammals. And in all cases, recognition of non-self by NLRs leads to oligomerization and ultimately to programmed cell death. So we're talking about an evolutionarily ancient defense strategy that's conserved across the tree of life. So NLRs can function in many different ways. In some cases, one individual NLR protein can mediate both pathogen perception and downstream immune signaling, such as SAR1, for example. This is what we term a singleton NLR. And then in other cases, these two functions are uncoupled into two NLR proteins, with one NLR protein acting as a sensor, sensing effectors, and then cooperating with a downstream helper NLR. And then these paired NLR configurations can become even more complex with multiple different sensors cooperating with multiple different helpers, and this is what we term an NLR network. We've heard about a bit about some of these in the previous talks. So in the Kamoun lab, we're very interested in understanding how paired and network NLRs evolve and how they function. Specifically, we've done a lot of work over the last few years on this particular NLR network, the NRC network, which is found in elastoid plants, mediates immunity against diverse pathogens. So it's composed of this layer of sensor NLRs, all of them recognizing effectors from different pathogens. And then these sensor NLRs signal through this layer of downstream helper NLRs, the NRCs, in a partially redundant manner. So we think that this redundancy at the helper NLR level provides the immune system a lot of robustness because the sensors have different immune pathways that they can activate immunity through. So the main question that I've been trying to address over the last few years is how do paired and networked NLRs get activated? We know a lot about how singleton NLRs activate, but we comparatively knew less about how paired and networked NLRs activated. And the system that we decided to use to address some of these questions is the specific sensor helper NLR pair from the NRC network. So it's a sensor NLR RX, which is a resistance gene that confers resistance against the against PVX by recognizing its code protein. So following code protein recognition by RX, RX can activate immunity via NRC2. And what we've been doing is to express this system transiently in Nicotiana benthamiana and then performing a variety of assays that involve biochemistry, cell biology, and structural biology, and use this system to ask and answer a lot of questions about how this NLR system works. And this led us to establish what we term the activation and release model. So what I'm showing here on the left is for comparison the first model for NLR activation established for SAR1. So as I told you earlier, SAR1 recognizes an effector, oligomerizes into this pentameric resistosome, and inserts itself into the plasma membrane. In the activation and release model that we've kind of fleshed out over the last few years, what we have is sensor NLRs recognizing effectors, and then following effector recognition, signaling through these downstream helper NRCs. And it is the NRCs that ultimately go on to oligomerize and form these resistosomes that insert into a plasma membrane. And the important thing here is that the sensor NLRs are not part of the final resistosome, hence the name activation and release. So there are differences in how paired and singleton NLRs activate. They have distinct activation mechanisms. Interestingly, if we focus just on SAR1 and NRC2, there are some similarities. In both cases, you have an NLR that's transitioning from low molecular weight to high molecular weight complex. But even in just looking at SAR1 and NRC2, there's already some differences. I'll tell you a bit about some of these differences that we found today. So I'll start with some of the differences we found for the resting state. And to tell you that story, I guess essentially we've always been interested in gaining any sort of structural insights really into how this NRC network operated. And we were very lucky that we were able to establish this pipeline that we've termed IPM here at TSL, whereby we express proteins of interest, epitope tagged proteins of interest in leaves of Nicotiana benthamiana. We harvest around 20 grams of agroinfiltrated leaf tissue, and then we do a preparative immunoprecipitation, and we then elute native protein purified straight from the plant that we can use for cryo-EM-based structural determination. And we subjected NRC2 to this pipeline, which allowed us to solve the structure of resting state NRC2, which unlike SAR1, which as I mentioned earlier exists as a monomer in its resting state, the structure of NRC2 revealed that it exists as a homodimer in its resting state. So it's two NRC2 protomers that are already coming together even before immune activation. And these two NRC2 protomers are held together by an extensive interaction interface that involves interactions mainly between the NBARC and LRR domains. So this was very surprising. It was the first instance of a resting state NLR reported to be in a homodimeric conformation. So we were wondering, why is NRC2 forming this homodimer? Why is it not a monomer like SAR1? What is the advantage of forming this homodimer? And to address that question, we decided to mutagenize this homodimerization interface and to see if interfering with dimerization has any effect on NRC2's activities. So again, we leveraged Nicotiana benthamiana. In this case, we performed cell death assays. Essentially, when you activate NRC2 in leaves of Nicotiana benthamiana, you get these necrotic lesions that are indicative of NLR-mediated signaling and immune activation. So we tried to activate wild-type NRC2 as well as all these homodimerization interface mutants to see if lack of homodimerization affected their signaling. So these are just the controls. When you have RX and NRC2 without the effector, you have this very healthy leaf tissue shown here on the top left. When you have RX and NRC2 with the effector, activation occurs, and then you have these necrotic lesions that you can see here on the top right. And what's really interesting is that for several of these homodimerization interface mutants, the NRC2 was now constitutively active, so it became an autoimmune variant, as you can see here on the left. So evidently, homodimerization seems to be important for autoinhibition and immune homeostasis for this NLR, this particular NLR. A stable homodimer formation prevents spurious activation and prevents autoimmunity. If we destabilize this homodimer by introducing mutations, you get autoimmune phenotypes and you get activation when it shouldn't happen, basically. So beyond contributing to autoinhibition, I mentioned earlier this network signaling architecture potentially confers robustness by having these redundant helper nodes downstream. We're also wondering how the dimerization of these helper nodes could be contributing to robustness in the immune system. So an observation that I didn't tell you about earlier is that we already knew that these helper NRCs preferentially self-associate. So when you have a co-immunoprecipitation experiment with NRC2 and itself or other NRCs, NRC2 preferentially associates with itself and not with NRC2 or NRC3 or ZAR1, which was included as a control. And now that we had this homodimer structure, we were wondering if this structure could give us some insights as to why this preferential self-association happens. So we looked at these interaction interfaces in a bit more detail. And we asked the question, have these dimerization surfaces or interfaces diversified throughout evolution? So the first thing that we did was to establish this data set of around 1,000 NRC sequences extracted from around 123 solanaceous genomes. And we built this very nice tree. You can see that the different NRCs cluster into these very nice clades with the NRC2 clade here on the right. And then we started looking at the dimerization interfaces throughout these different NRC clades. So just for comparison, these are some well-characterized, highly conserved motifs that are found in NRCs and even in other NLRs, non-NRC NLRs. So these motifs have been already shown to be very critical for NLR function, and you can see that the lower plots suggest high degrees of conservation. But if you compare those conserved motifs to these motifs, so these are the motifs for the dimer interaction interfaces, there doesn't seem to be any evidence for conservation. It actually likely seems like they're more diversified across all NRC helpers. So this was when we looked at across all 1,000 NRC sequences. But what was really cool is that when instead of looking at across all the 1,000 NRC sequences, we started looking at these dimerization interfaces within each of the NRC clades. Now we did see strong evidence for conservation. So the dimerization interfaces are quite conserved within each of the NRC clades. And they're not only conserved, they also feature unique polymorphisms within each clade that we think might be contributing to isolating the different NRC clades from each other. And why would evolution lead to this kind of isolation of the different NRC clades from each other? Well, essentially, what we think is that, well, first of all, we have a network signaling architecture that is not only redundant, but these redundant nodes are also insulated from each other, like I mentioned. And the reason why we think that could be happening is related to effectors. So effectors can activate NLRs, but other effectors can also suppress NLRs. So we think that by having these helper nodes that are not only redundant, but also insulated from each other, this might make it more difficult for pathogens to evolve to suppress these helper nodes. And I also want to tell you, so that's a bit about the resting state of these NRCs, but I also want to tell you a bit about the activated state of some of these NRCs. So there's diversity in the active form as well. So we use the same IPEM pipeline, this time focusing on the RX and CP activated state of the helper NRC2. And this allowed us to solve the structure of the activated NRC2 resistosome, which unlike SAR1, which forms a pentameric resistosome, we found that the activated NRC2 resistosome forms a hexamer upon activation. So yes, again, there's diversity both in the resting states and in the activated states. So we finally obtained structural confirmation of this activation and release model that we had proposed a couple of years ago based on biochemical evidence. So we have confirmed that the sensors are not part of the activated hexamer. The sensors are merely converting these helper homodimers into helper hexamers, following effector perception. And why is this important? This is important because initially, before we had any of these structures, actually paired NLR systems are also found in mammalian innate immunity. And in mammalian innate immunity, both the sensor and the helper come together to form a sensor-helper heterocomplex. So we were actually thinking in the beginning that this might also be the case for plant paired NLR systems, but it turns out that plant and mammalian NLRs exhibit distinct activation mechanisms, which we thought was pretty cool. So just to wrap up, I told you about this activation and release model that we've been able to build over the last few years with work from several different labs, including Sofian's lab. I've told you about how this NRC network features this redundant signaling architecture, which is where the helper nodes are not only redundant, but also functionally insulated, potentially to prevent pathogen suppression. I've shown you these two new structures that we've gotten for the resting and activated states of NRC2. So we're starting to expand our current understanding of the NLR immune receptor structural landscape. So there's more diversity. than we've previously anticipated, both for the activated states, but also for the hugely understudied resting states of these NLRs. I think a lot of people were more interested in the resistosome than in the resting state, but there's a lot of cool stuff going on even before activation, in my opinion. But the thing is, we're convinced that that's just the tip of the iceberg, really. I mean, there's a lot of diversity still left to uncover, so we're very excited to try and uncover it. And just to wrap up, I'd like to say that I think this work very nicely exemplifies how by combining structural biology, phylogenomics, and functional analysis, kind of this triad that we've termed IS-MPMI, I think it's a very powerful way to generate experimentally testable hypotheses and to address fundamental questions in the field of plant immunity. And with that, I'd like to thank our collaborators, a lot of people that helped us with the structural biology, Sofian, my supervisor, Selva, Madu, and Amir Ali, who contributed to different aspects of this project, as well as all of the Kamun Lab and our collaborators. And with that, I'd like to take questions. Yeah, thank you very much.
Thank you, Mauricio. I think we have one question from Jean. So her question is, can we think of helpers as specific chaperones?
As specific chaperones. I guess to me, chaperone kind of implies some sort of contribution to the stability of the protein that's acting as a chaperone, or to the folding of the protein that it's acting as a chaperone to. What I can tell you that the way that I'm conceptualizing at least the NRC helpers at the moment involves the NRC's kind of monitoring the status of the upstream sensors. And I didn't have time to talk about that story today, but we have a very nice story that we published last year on that. Basically, what we think is going on is that the sensor LRs have degenerated and lost a lot of their functionality. They are unable to form resistosomes. They are unable to trigger cell death. And they're in a way almost acting as if they were decoys for the effectors. So, and essentially the helpers are monitoring the status of these upstream sensor LRs. And then activating after perceiving certain conformational rearrangements in the sensor NLRs. That's how I conceptualize it, at least.
Great. And Shohini says, awesome talk. I was wondering if you have looked or considered looking at any signals of selection, balancing positive neutral among your NRCs.
Yeah, so that's a very good question. We are about to publish a study on that. And we do find all sorts of different things going on. I think it depends really on which region of the NLR we're looking at, but we do see evidences for different types of selection pressures. The helper NLRs in general tend to be quite conserved, but in the case of the sensor NLRs, there's very strong coevolution with the effectors. So in that case, there's more interesting stuff going on. I think the helpers tend to be more conserved because all they're really doing is monitoring the sensors, but even then, in some cases, we do see evidence for some degree of diversification because at the end of the day, the helpers are co-evolving with the sensors as well, right? As new sensors appear, the helpers need to kind of start to co-evolve with them. And also now we more recently observed that actually, because some of these effectors are targeting helper NLRs to suppress them, there's also co-evolution at the level of helper NLRs with their suppressors trying to evade suppression, not necessarily trying to gain recognition, but trying to escape from effectors. So there's a lot of very interesting things going on, evolutionarily speaking, across all levels of this network, I would say.
Thank you.
Great. And Adam asks, there are of course smaller numbers of proteins in the NRC subclades. How does degree of conservation at interface domains compare to conservation at random sites in the proteins?
Sorry, can you read the question again? So, I think you're muted, Yumina, sorry.
I can
My
jump in
bad.
because maybe I can make
Yeah.
clarify. So you see this really striking conservation at the interface domains within NRC subclades, right?
Mm-hmm.
That differentiates, and you propose isolates them one clade from the other. But because those are smaller numbers of proteins within those subclades, we could just by chance have less diverse sequences within, across the protein anyway.
Yeah.
So is the relative degree of conservation at the interface domains greater than, the degree of conservation at random sites in the proteins? Is it significant, in other words?
Yeah, so I think basically if you look at those dimerization interfaces, the degree of conservation that we're seeing looks more akin to the conservation that you would see at highly conserved motifs like the MHD motif or the N-terminal mana motif. and it's more conserved than random portions of a protein. And just to add a bit more to that, we do have functional evidence. So we have done experiments where, I mean, you can see that, you can read that up in the PLOS Biology paper that we published on this topic, but we did some experiments where we swapped these regions, we mutated these regions, and we do see that that contributes to isolation of these helper nodes. So we have some functional evidence as well. Yeah. Thank you for the question.
All right, and Marcelo asks, does resistosome formation in animals also lead to cell death? Do you know if there is do you know if immune function is conserved there, or would it be part of
Yeah, so
other signal transduction processes?
So in the case of mammalian inflammasomes, ultimately it does lead to cell death, although the mechanisms are different. So in the case of plant NLRs or plant CC type NLRs, at least, the resistosome itself is most likely triggering the cell death by puncturing the plasma membrane, amongst other things. In the case of the mammalian inflammasomes, so the inflammasome that I showed you, which is an NLRC4 inflammasome, acts more of like a signaling scaffold, which leads to caspases activating, and then these caspases activate gasdermins. And then these gasdermins are the ones that are triggering the cell death. So in the case of the inflammasome, the inflammasome is not the direct executor of cell death, but it activates a cascade that eventually leads to cell death.
Great, thanks, Mauricio. There's one more question, but if you could answer in the chat, that would be awesome. We're going to move on to our next speaker.
Thank you.
So, thanks. Our next speaker is Jade Blue. She is a postdoctoral scholar at the University of Dundee, and her fun fact is, at least according to her mom, she was the first baby born in London in 1995. So that's kind of cool. Jade is going to be talking about effector cooperation and plant-aphid interactions. Go ahead, Jade.
Perfect. So firstly, thank you for giving me the opportunity to talk about some of our research today, and thank you all for coming. So yeah, today I'm going to talk a bit about some of the work we're doing in the lab on aphid salivary proteins that we call effectors and how they might be working together during plant-aphid interactions. So just, there we go. Brief introduction to myself. So my name is Jade. I'm a postdoc based at the University of Dundee, which is at the James Hutton Institute in Dundee in Scotland. And I'm interested in plant pest pathogen interactions, but I've got a specific interest in plant-insect interactions. So in the lab, we work on aphids. So these are phloem-feeding insects. They colonize plants quite extensively. They produce clonally during warmer months. You can see here on the left on a mustard plant, and also some unwanted aphids on our stock plants in our greenhouse. And they cause direct damage through feeding, but they also cause indirect damage through honeydew secretion while they're feeding, which attracts secondary pathogens. And they also transmit viruses, which contributes even further to crop loss. So control of aphids is particularly challenging because we have limited genetic crop resistance, which means we have a reliance on pesticides. And due to an increase in pesticide resistance, coupled with our need to decrease our use of pesticides based on the negative environmental impacts, we need to identify more novel and sustainable methods of resistance. And in the lab, we're looking to understand more about the molecular dialogue between aphids and their plant hosts as a way to do so. So as I mentioned already, aphids are phloem-feeding insects, along with others you might be familiar with, such as whiteflies and leafhoppers. And they feed using these specialized mouthparts called stylets. And when they're probing and feeding, they secrete saliva into plant cells. And this saliva contains small molecules, including small proteins. And several of these small proteins that we call effectors have been found to promote susceptibility to aphids. And they've been found to do so by interacting with host proteins and disrupting host processes. So in the lab, our overarching question is, how do aphids manipulate host processes, particularly through effectors? And what some of us are working on at the moment is whether these effectors work together to do so. So what led us to hypothesize that effectors might be working together is based on work previously published by the lab, where they looked at the effector organization in the genome of different aphid species. And you can see with the red and the yellow lines here that in all the species they looked at, they identified a pair called MP1-like and ME10-like that were always found localized next to each other in the genome, whereas the surrounding genes were quite different. So they looked further into Myers persicae, which is a generalist aphid with a broad host range. They looked at the expression profile of these genes in the body versus the head, where the salivary glands are, and under different feeding conditions. So here you're looking at the orange and the yellow line, and you can see that under all the conditions that they tested, the expression profile of the two aphid genes are very similar. So what are MP1 and MP58? So they are small proteins that are found in the saliva and salivary glands of Myers persicae. Their structure is not known, but their predicted structure is that they're mainly made up of alpha helices. And with MP1, it's thought to, part of its role is thought to promote susceptibility through interaction with a trafficking protein called BPS52, which I'll speak about a bit later. And with MP58, very little is known about it, but a homologue in the potato aphid, ME10, is known to interact with a 1433 protein in tomato, which is a family of proteins that are known to be important for immunity. So it is known the effect of these effectors when you overexpress them in plants and their effect on aphid performance. So with MP1, when you express it under a 35S promoter, so constitutively, you actually don't see a difference in fecundity, so the amount of offspring they have, whereas if you express it specifically under a phloem-specific promoter where the aphids are feeding, you do see an increase in fecundity. What's quite interesting is with MP58, on the other hand, when you express it both constitutively and under a phloem-specific promoter, you actually see a reduction in fecundity. So with all of this, we wanted to look into what these effectors might be doing together in plants. So I first looked at where these effectors localize, when they're expressed individually, and whether this changes when they're expressed together. So you can see with MP1, it's predominantly localized to the nucleus with a weaker signal in the cytoplasm. When you express MP58 on its own, it's predominantly expressed more nuclear in the cytoplasm. But what's quite interesting is when you co-express these in benzimiana, I should have mentioned, these MP1 relocalizes to the cytoplasm and you no longer see it present in the nucleus, whereas MP58 stays the same. So it suggests that there's an association between these two effectors in plants in the cytoplasm. So I next wanted to see if they also interact with each other. So I co-expressed MP1 with MP58 or with a control, MIT-GUS or GFP. And when you pull down MP1, I also pull down MP58. So it suggests that these proteins interact in plant cells. And I'm not showing the data, but we also looked at this in vitro with recombinant proteins and also were able to co-purify them together. So it suggests that this interaction is direct and it's not necessarily facilitated by something in the plant. So as I mentioned at the beginning, this effector pair that was identified is conserved between different aphid species. So I also wanted to look at whether this interaction is conserved in the cereal aphid, which has a different host range to Myers persicae. I'm not showing the data, I'm showing a schematic for this for the sake of time, but we also saw an interaction between the cereal aphid homologues. What's quite interesting is when I did a co-IP experiment with looking at the interspecies interactions and whether that's possible, I wasn't able to detect an interaction. So it suggests that maybe this effector pair has evolved separately in their respective aphids for different functions in different hosts. So after identifying that this effector pair forms a complex, we wanted to look more into how these effectors might be working together during aphid infestation and what they might be targeting. So as I mentioned earlier, MP1 targets a protein called BPS52. And BPS52 is a member of the GARP complex, which is important for tethering vesicles to the Golgi apparatus. And it's highly conserved in eukaryotes. And so the structure in plants, as far as I'm aware, hasn't been resolved, but so this is a predicted structure that a postdoc in our lab, Ronan, made, and it suggests that as is with other species, that the GARP complex interacts with this alpha helix and terminal region. So MP1 can interact with BPS52 in a host-specific manner. So it interacts with host homologues such as Arabidopsis and potato, but it doesn't interact with non-host homologs such as barley. When you overexpress BPS52, you see a reduction in aphid fecundity. And what's quite interesting is normally you see expression predominantly in the stems and the flowers of plants. But when you put aphids on the plants, you see degradation of BPS52. So it seems like it is an important target for aphids. So I first wanted to test whether MP58 does also interact with BPS52 and whether an interaction is dependent on MP1. So I expressed BPS52 with MP58, the aphid protein, with either MITGUS as our control or MP1 to see if it interacts in the absence and presence of MP1 or with our controls. And when you pull down BPS52, you can see here that you can pull down MP58 with just MITGUS, so in the absence of MP1, but you also can pull down MP58 with MP1. So it suggests that MP58 interacts with BPS52 independently of MP1, and also that MP1 doesn't disrupt the MP58 BPS52 interaction. And I'm not showing the data, but we've also looked at it the other way around and we see the same result. So we wanted to look more into where MP1 and MP58 might be targeting BPS52 to understand more about how they could possibly disrupt its function. So Ronan modelled the two aphid effectors MP1 and MP58 with Arabidopsis BPS52, and his models predicted that they interact at this N-terminal alpha-helix region, which is the region that's important for the interaction with the GARP complex. So Namami, a PhD student in the lab, designed constructs where you have the full-length Arabidopsis BPS52, which we know interacts with MP1 and MP58, the non-host barley BPS52, which we know doesn't interact with MP1, and chimeras where you swap the N-terminal region of AT BPS52 with barley and vice versa. And so then with these constructs, I did a co-IP, and there's a lot of Western blot data here, so feel free to just pay attention to the schematic at the top. But essentially, I expressed these BPS52 variants along with MP1 or along with MP1 or just GFP, and that's on the left, and on the right is the same experiment, but just with MP58. And when you pull down Arabidopsis BPS52, you can also pull down MP1, and when you have the variant with just the N-terminal region of Arabidopsis BPS52, you can also pull down MP1, but we don't see any interaction between barley, which we know, or the variant with the barley N-terminal region, and we see the order of the lanes are a bit different, but we see the same with MP58. So it suggests that this is indeed the region that is important for association of BPS52 with the aphid effectors. So our current hypothesis is that when, in the absence of aphids, this N-terminal region of BPS52 is free to interact with the GARP complex and function as normal. But when you have when you have aphids on the plant and the MP1 and MP58 is secreted into the cells, they bind to this N-terminal region and potentially block binding to the GARP complex and downstream signaling. But we're still in the process of validating this mechanism. And we still have some other questions to answer. So I've shown you that these proteins interact independently with BPS52, but we haven't actually confirmed whether they interact as a complex, and that's something I'm working on at the moment. And we also aren't sure if the MP1 and MP58 complex formation regulate any BPS52 associated activities, and that's something we're also working on at the moment. So just to summarize what I've told you today, or what I've shown you today, I've shown you that aphid effectors form complexes with each other. I've shown you that this MP1-like and MP58-like complex is conserved in different aphid species, and that MP1 and MP58 both share host protein targets, but whether they are both important for disrupting function of VPS52 we still need to determine. And this is quite a novel feature of effector biology, I guess, in plant insect interaction. So it's effector complexes have been shown before in bacterial pathogens and fungal pathogen effectors, and there's this term meta-effector where effectors regulate the activity of other effectors, but we're not sure exactly how common this is. We don't know where these effectors form, whether it's in the salivary glands or once it's secreted into the plant, and whether this is important for the regulation of these effector activities. And also something I'm particularly interested in looking at is whether complex formation is important in diversification of host targets. So you might have your individual specific targets, you might have shared targets as I've shown you today, but you also might have a novel, when these effectors form a complex, do they take on novel functions and target new host proteins, potentially to increase that resilience against any plant defenses. So with that, I would like to thank the lab. So I've shown you a lot of the structural predictions from Ronan, but Tom is a postdoc in the lab who's done a lot of in vitro work related to this. And Amami's a PhD student who's done a lot of cell biology, so I also didn't have time to show you their data today. But yeah, so I also the rest of the lab and the James Hutton microscopy facility who have helped me a lot, and thank you all for your time. And happy to take any questions.
Thanks so much. We have a question from Jean. She asks, could you remind us why binding to VPS52 results in better aphid performance? What is it doing?
Binding of the effectors to
Yes, to
VPS52.
the VPS52.
So we haven't actually established the direct link between the effectors and VPS52. So we know that separately MP1 can increase aphid performance, and we know that when you overexpress VPS52, it decreases aphid performance. So it's currently hypothetical. When aphids are on the plant, VPS52 is degraded, but this also hasn't been directly linked to MP1. We're hypothesizing at the moment that potentially MP58 might play a role in that. So maybe if you have them together, but we're doing those experiments at the moment to look at VPS52 degradation, but we're still trying to establish that.
Thanks. And Maurizio asks, he says, great talk. Are MP1 and MP58 encoded in close proximity in the aphid genome? Do you think that by leveraging specific structural features or potentially residues at a potential MP1-MP58 interaction interface, whether that will allow you to fish out other effectors that are working as complexes?
So yeah, that's a good question. Yeah, they are encoded in close proximity in the genome. They kind of share similar promoter regions right next to each other. They're right next to each other, and the promoters are very similar. So we think they're encoded in close proximity. And we're also, so Tom's doing a lot of the in vitro work related to this, and he's also trying to uncover the structures and how they also might be targeting host proteins as well. So yes, we do think that, and we're trying to work it out at the moment.
Thank you.
And Jean has her hand raised.
Yeah, just to follow up, I guess what I was trying to get at is you see differences in the VPS52 level, whether it's direct or not, but why might that result in a change in aphid performance?
Well, because we think that VPS52 is involved in protein trafficking. By disrupting that, you could potentially disrupt any proteins packaged in vesicles that might be maybe increasing defense towards aphids. That's a potential hypothesis.
Okay, just to like zoom out again.
Yeah.
Reminding us why the physiological relevance. Thank you.
And Adam has a follow-up to Mauricio's question. Have you looked specifically at sequence variation across orthologs of the effectors that you're looking at?
Yeah, so They range between, I guess, 50 to 60% similar for both MP1 and MP58 homologs, but their structures are very similar. They look, to me, as not a structural biologist, look more similar than the sequences, but their shapes are quite similar.
All right. Well, if we are done with questions, I think we'll move on to the next talk. Thanks so much, Jade. So our next speaker is Shohini Guha, and she is a postdoctoral scholar from Pennsylvania State University. And her fun fact is that her 3-year-old just made her very first friend, and she is much more excited about it than her child is. So that's exciting for all of us. Thanks, Shohini, for sharing that fact. And her talk is titled Mutations in Legume Symbiosis Genes Create a Complex Selective Landscape for Natural Genetic Variation in Rhizobia During Multi-Strain Inoculations. So go ahead, Shohini, whenever you're ready.
Can you see my presentation? Is it visible to everyone? Okay.
Yes.
Well, I would like to start by saying thank you to IS-MPMI for giving me this opportunity to showcase my research today. As already been mentioned, my name is Shohini Guha. I am a postdoc in the Liana Burkhardt lab at Penn State, Department of Plant Sciences, and I study legume rhizobial coevolution. So throughout my talk, I would like to address how sensitive rhizobial fitness is to host genetic perturbations. And I would also like to address which rhizobial genes actually influence these strain fitness shifts across these host mutations. And then I would like to end my talk by giving a broader perspective of how my work adds to the ongoing research that is happening in this field. Now, I have divided my talk, I have categorized my talk into several subsections. And as I advance, I would walk through each of these subsections. So starting with background first. So plant microbe interactions can be broadly categorized into beneficial and harmful. And I have listened to several excellent talks that address the pathogenic side of these associations, whereas I work on the symbiotic interactions that a plant establishes with microbes. So my area of research is biological nitrogen fixation. So biological nitrogen fixation, it is a symbiosis. It's a process by means of which certain plants called legumes, they enter into a mutualistic association with a group of bacteria called rhizobia. In this process, there is an extensive molecular crosstalk that happens between the plant and the bacteria, as a result of which the bacteria colonizes some specialized structure in the roots of these legumes. These are called nodules. And within these nodules, there happens to be an exchange of resources. The bacteria provides the plant with some assimilable forms of nitrogen, whereas at the same time, it receives carbon and shelter from the plant. So this is a very good example of what a symbiosis actually looks like, and it has been thoroughly researched. But if we actually look into the literature of biological nitrogen fixation, we would see that the research has been heavily skewed in the direction of the plant. We have been more concerned how to fine-tune this mechanism to maximize plant benefits. But it has been poorly understood, or I would say understudied from the perspective of the microbe. And it is important that we care about this, because since we are now in the era of sustainable agriculture, we are vastly promoting sustainable agriculture. When we are trying to articulate or when we try to designing microbial inoculants, in most of the cases, we find that they fail to perform optimally. And these problems are some common problems like failure to compete with the resident soil microbes, or that leads to a competition problem that leads to their failure to establish. Eventually, that leads to mutualism breakdown and ultimately suboptimal biological nitrogen fixation, which eventually impacts the plant benefits. So the bigger question here is there a way to like maximize the fitness, the performance? Is there a way to improve the performance of these microbes? And that is where understanding and knowing how we can, improve microbial fitness comes into play. Before going any further, I would like to introduce the concept of fitness landscape. So fitness landscape, it is a conceptual model that actually connects phenotypes to fitness. In a given environment, the best combination of traits lead to optimal fitness. And over time, we expect the whole population within that environment to evolve to that state of maximal fitness. But in real world, environments change. And so the fitness landscape of the population within those environments also change. And in the world of host microbe interactions, inter and intra-host genetic variation, they create a very dynamic fitness landscape for the microbes in those environments. And we have very little grasp on the magnitude of this variable selection. My model, I study with Medicago. Medicago is a model plant for studying nitrogen fixation. It showcases the symbiotic relationship between plants and rhizobia. And research on biological nitrogen fixation genetic relies heavily on Medicago because of the ready availability of resources. There are like tons of host genetic lines and established GWAS panels, sequence resources. So we have a lot of resources based on Medicago. So it is a very good model system to study nitrogen fixation, biological nitrogen fixation, and at the same time, rhizobial fitness. The beauty of this system is there exists a vast scale of host variation between Medicago species, even within a particular species, Medicago truncatula, and even within mutant lines of a particular genotype. So existence of these variations, they offer opportunities to explore how host genetics will influence fitness of rhizobia. And this is one of the many projects the Burkhardt Lab has been investigating, where we try to understand how variation between species, within species, and variation at the level of individual mutations in a single genotype would impact rhizobial fitness. I specifically worked on the last bit, like I explore how mutations in individual host genes would impact rhizobial fitness. Root nodule symbiosis, which eventually leads to biological nitrogen fixation, has a very complex underlying signaling. And if you look at the genetic components that regulate that, we can see it is pretty large. We are still discovering genes, plant genes, which regulate biological nitrogen fixation, root nodule symbiosis, still date. But this huge resource, this huge information can be loosely clustered into 3 stages of symbiosis: the early, late, and AON. AON, it's the short form of autoregulation of nodulation. It is a signaling pathway that prevents the plant from making more nodules. So nodulation is a very energy intensive process, and there has to be a mechanism that should prevent the plant from like overexerting itself. So that's what AON is. In my work, in this research that I will be talking about, I have worked with these mutant lines. Each of these represents a single mutant line of Medicago, and the genes, you don't have to remember the names of the genes. These genes, they impact either the early, late, or the AON stages of symbiosis. So historically, how these genes were discovered. So we work in a single strain. We have been traditionally working in single strains with single strains. We expose a host mutant to a single strain. We score for binary traits like, does this mutation impact nodulation? If it does not impact nodulation, then we try to see whether the nodulation is functional or not. If the nodule that is formed is small and white, it means it usually means that the nitrogen fixation is suboptimal. Or if the nodule is big pink because of the production of a specific pigment that is only produced when the nodule is fixing nitrogen, then we say that it has a positive nitrogen fixation phenotype. And over the years, we thought that these phenotypes were universal, but then certain instances started emerging where we found that these phenotypes are not universal, and sometimes they depend on the strain that the plant encounters. For example, in this particular gene, NPD1, when you mutate this gene in Medicago, when it faces strain 1, it has got a positive nitrogen fixation phenotype. But on the other hand, when it faces strain 2, the phenotype is ineffective nitrogen fixation. So this brings us to a bigger question. Do host genes influence rhizobial strain fitness? In a realistic agricultural scenario, a legume at a time faces tons of bacteria. So how do these host genes influence rhizobial strain fitness? Before we start looking for answers, how do we measure rhizobial fitness? So we can measure rhizobial fitness. For example, if you're trying to see the rhizobial fitness, we're trying to measure the fitness of like 4 strains. We start by mixing those strains in known proportions, expose that strain cocktail to a certain plant, a certain mutant, re-isolate the population of bacteria from those mutants, and then estimate the frequency that is coming out of the plant. Once we have that, we take a ratio of the final frequency to the initial frequency, and the log of that value would give us the fitness of each of those strains in that particular mutant line. So using this logic, we will be using this logic to answer this question, like, do host genes influence rhizobial fitness? So in order to answer this question, we had an elaborate experimental design. So this experiment was completely done. I wouldn't say mostly, it was completely done by my mentor, Liana Burkhardt, and Gina Bledsoe, who was a previous tech in the lab. So what Gina did, she grew 86 Cynorhizobium strains individually, created a strain cocktail. She then exposed that strain cocktail to each of these host mutants and the wild type. Finally, she extracted the bacteria, re-isolated the bacteria from each of these mutant lines, did their pooled sequencing, the whole genome pool sequencing, inferred the strain frequencies, and then finally calculated the strain fitness. With this data in hand, we were now in a position to answer this question, like how sensitive is rhizobial fitness to host genetic perturbations? So what we did was, I will be showing a heat map here now where I have shown the relative fitness of each of these 89 strains along the x-axis, and I have mentioned each of the host genotypes along the y-axis. I would like to please take a look at the key here. Anything that is shown in red means it is enriched, it has got a high strain fitness. Anything that is shown in blue would indicate it has a low strain fitness. I would like to start small. I am here showing the strain fitness of 89 strains in the wild type plant A17 and in a mutant LAT-D in the same host background. As you can see, there exists certain differences between the strain fitness in the wild type as well as compared to the host mutant. When we look at the whole trajectory, we see that yes, there is variation in rhizobial fitness across the host mutant lines. Certain things become evident here. We see some clusters happening across the heat map, and these clusters are independent of the stage of symbiosis. We also noted that some strains were persistently maintained at high fitness, some were maintained at low fitness, but there was like 70% of the whole selected consortia which showed variation in their strain fitness as the host mutants changed. This observation led us to conclude that, well, extensive host by strain genome interactions is happening across the entire developmental trajectory of root nodule symbiosis. And when we had this data, we then asked the next question. Well, which rhizobial genes underlie this response to changes in the host genetic components? To answer that, we ran a GWAS, we ran a genome-wide association study on strain fitness shift. So what GWAS tries to do is it tries to statistically associate a particular trait to a particular SNP variant. And if that association is significant, it suggests that the SNP is influencing that trait. Now, for this talk, our trait is going to be the strain fitness shifts. And how did we get that strain fitness shifts? By simply subtracting the fitness of a particular strain in wild type from the fitness of the same strain in the mutant. So the resulting difference would give us the fitness shift of that particular strain in that particular mutant. That was the trait we used for running our GWAS. Before I go,
you have 2 minutes.
Okay, I'll speed up. So before I show you the candidate genes that came up through our GWAS, I would like to give a brief preview of what a cynorhizobium genome looks like. So it is a tripartite genome. It has got a chromosome and 2 megaplasmids with the genes responsible for symbiosis located on PSIM A. When we plotted the candidates that came out of GWAS, we saw that most of them were clustered on the PSIM A and PSIM B. Some of them seem to be shared across multiple host genotypes, whereas some showed up to be in just a single one of them. The former category was called pervasive, the later one was called limited effect gene. I would like to just explain how we did the categorization. For example, this particular loci on chromosome, it comes up as important based on its p-value rank in just a single host mutant, RDN1. This is a limited effect gene as opposed to these two loci, which were important across multiple host genotypes. So using this logic, when we looked up at the whole genomic landscape, we could see that there were some pockets of genes on PSMA which seemed to be like essential across multiple host genotypes. So these were the pervasive genes. We were interested to see what these genes were. And some of them were already known to influence nitrogen fixation, whereas some were novel genes. So that was interesting. So the take home was, yes, perturbing host genes do alter fitness landscape of rhizobia. And finally, using GWAS, we can identify novel genes that may have a role in influencing rhizobial fitness across multiple diverse host environments. And if you're interested, this work is currently up in bioRxiv. So you can read up if you're further interested. So bringing to the next steps, how does this add up? So now that we have identified the genetic drivers underlying rhizobial fitness in mutant lines, the lab is extrapolating to additional genetic scales like different genotypes of Medicago truncatula or different species of Medicago. And as we do so, we would be in a position to create a holistic overview of the genetic drivers responsible for maintaining strain fitness shifts across the different host environments. And this information can set the stage for improving microbial inoculant performance. So with that, I end my talk. I thank my funding agencies. And lastly, and not the least, the lab itself. It was a team effort. Thank you.
Thank you, Shohini. If anyone has questions, please type them in the chat or feel free to raise your hand. I can start with one question. So do you think the genes that you discovered to have sort of the pervasive effects be used to broaden host range of rhizobia?
That would depend, because the genes that we found out, they had like a wide range of functionality. Some of them were like core metabolic genes. Some of them were like the motility genes. So we expect that they might or might not influence rhizobial competitiveness, rhizobial fitness. But if they do, then the mechanisms would be widely different in different host mutants.
Thanks. Jean, please go ahead.
Oh, since you had to rush a little bit at the end, maybe you could tell us a little bit about those novel things you found and what's the prospect of taking advantage.
So the novel genes that we found out, they were mostly like hydrolases and NADP binding proteins, which so the NADP binding proteins, they usually act like electron carriers for several biochemical pathways. We are actually in the process of validating those genes. We are actually in the process of creating mutants of those genes and checking out if they do have any, if they do impact rhizobial fitness, and if they do, what is the mechanism underlying it? But those are some exciting answers that we are waiting to come up. We are just in the process of validating them. Yes.
Mauricio asks, maybe off topic, but is there any evidence of intraspecies variation for these host genes?
So yes, we already have data for that. We did a population genomics. We have done the comparison with 202 accessions of Medicago. We have the data for the genetic variations. What we found that, and that this has already been published, what we have found that there exists no strong signal for positive or negative selection. It is mostly balancing selections that we find across the entire database of genes, of host genes. So I'm not sure what's going on there, but that's what we have found. There is variation, to answer your question, yes. But as to signals of selection, well, it was mostly balancing.
Thank you.
Thanks, Shohini. And I think that about wraps up the last speaker. So I'll hand it over to Adam and Katie for the breakout sessions.
Great. Well, thanks everyone. Thanks for the great talks to all the speakers, and thanks Yumino and Linan for moderating the session. Before I hand it over to Katie for the breakout sessions, I have just two more quick announcements. Be sure to tune in next week for the second part of this showcase. Another great lineup of 6 exciting talks. Also, just a reminder that abstract submission for the July Congress in Cologne, Germany closes on March 11. You see that there. I'll also put the link in the chat if you don't have time to screenshot that Q code. So be sure to get your abstracts prepped up and submitted, and then registration will be open about the same time, and you'll have more time for that. I think that's it. Katie, did you want to guide us now into the breakout rooms? These will be sort of, open discussion facilitated in each room by a member of the board. So we welcome you to choose one that's of interest. You can also go between rooms during the remaining 40 or 45 minutes that we have. Anything to add there, Katie?
You took my job from me, Adam. So thank you very much.
Sorry.
Yes. No.
Okay.
I here momentarily, I will be opening the rooms, and all attendees will be able to choose which breakout topic that you would like to speak, you would like to have a discussion about, or hear about.